So in this experiment, what we did is we took a leaf. Okay, we took a leaf and we took a beaker. Oh, okay, we took a leaf. This is a test tube. Okay, this is the test tube. This is a leaf. Okay, and here in the test tube we have a cotton soaked of KOH solution. Okay, and this is the leaf. This is the variegated leaf. This portion is at the atmosphere, and this is in the test tube where we have kept the KOH soaked cotton. Okay, so what will happen in this case? There will be, and it is exposed to air. Okay, it is exposed to air. Okay, and here it is KOH solution. Okay, so in the presence of what we are going to do in the setup, when the presence of starch or starch will there, okay, then it will show that here it will the process of photosynthesis will take place, and here the process will not take place. Why it's going to happen? Because in the beaker, in the test tube, we had what? We had the KOH soft cotton. It will absorb all the CO2. So what will happen? Process of CO2 which traps the sunlight will not be able to trap it, right? And because of it, it will not be able to perform the process of photosynthesis. So in this experiment, this is done, and the next experiment is showing that it is interesting to learn about the simple. This experiment was done by Joseph Priestley, okay, in 1770. In 1770, a series of experiments were revealed, okay, in which we noticed the growth of green plants only. Priestley did an experiment in which he observed that a burning candle in a closed space, a bell jar, soon gets extinguished. Okay, so what he did is, look at the picture. This is a very interesting experiment, by the way. You will love it. So what is it happening here? He took a bell jar. This is a bell jar, okay? This is called as a bell jar. He took a bell jar, and what he did in the bell jar, he take a mouse for his experiment, and there is a candle. Okay, when he put the mouse without this plant, then what happened? Due to CO or accumulation of CO2 in the bell jar, the rat died. It got died, and the candle also got extinguished. So why it happened? Because the accumulation of CO2, because of the accumulation of CO2 it happened. By this, because somehow the rat was doing the was polluting the air, and somehow there was no oxygen, there was absence of oxygen, due to which this happened. But in the second case, what we did, we take a plant of what? This is the plant, mint plant. This is the mint plant. Mint mint plant. Okay, we took a plant, mint plant, and we took this candle and this mouse. Here it will remain the same. Look, the rat is alive, the candle is also burning. Okay, because our this plant has produced O2, and there was not the accumulation of CO2 in the jar, sorry. There is O2 present. Okay, and due to the oxygen, the candle remains burning, and the rat also didn't die. Okay, so this was a pretty cool experiment. Next experiment was done by Engine House. So what he did, Engine House is his name. Okay, Engine House is an elegant experiment with an aquatic plant, and when he put the aquatic plant in the bright sunlight, some bubbles were formed, and the bubbles were what? These were the bubbles of O2, okay, and were formed around the green parts of the plant, while in the dark part it didn't form. Why it didn't form in the dark part? Because there was no presence of O2, na? And because of O2 only they formed, right? So it shows that sunlight is important, right? So he did an experiment in the aquatic plant. Okay, he identified these bubbles that to be of oxygen. These were the bubbles of oxygen. Okay, so what he concluded, this is only because the green plant of parts of the plant only releases oxygen. The green part of the plant can only release oxygen, whereas the plants which like the dark side of the plant, dark part of the plant will not produce it oxygen because they did not go under the process of photosynthesis. Okay, this experiment was done by Engine House. In which year? Jan Engine House did it experiment. The year of experiment is not given, but the time is given, like 732. He was there in 732, 1799. Okay, this was the lifespan of him. Next experiment, Julius von Sack. Next experiment was done by Julius. Okay, so what he did the experiment here. The experiment date is given, right? It is 1854. 1850. Yeah, 1854. ठीक है, तो Julian von Sachs what did? They provided evidence for the production of glucose. He tell that when plants grow, they will produce glucose. Glucose is stored as the form of starch in the plants. Okay. Later he studies that the green substance in the plants, chlorophyll, is located in the special bodies which are called as chloroplasts. Later he identified that. Okay. So this was done by Julius von Sack. Next we have T.W. Angelman. Angelman. We have T.W. Angelman. So what he did, we have T.W. Angelman. What he did, he used a prism. He used this here also. We have the lifespan of him. He used a prism. He split the light into spectral components and then illuminated a green algae, which is cladophora. Used cladophora and it placed in the suspension of aerobic bacteria. So what had happened? The bacteria were used to detect the site of O2, that where O2 gets evolved, that in which portion O2 gets evolved. So he observed that the bacteria accumulated on this area where they had the O2 presence, right? So this shows that the bacteria only accumulated mainly in the region of blue and red region of the split spectrum. So this shows that there was the high accumulation of O2. That's the reason why it is done in the red and the blue region only. Okay. And it roughly also resembles the absorption of spectra of chlorophyll A and B. By the middle of 19th century, there were some like this equation, the empirical equation. This is the empirical equation which represents the total process of photosynthesis for oxygen evolving. Like we take CO2 from the surrounding with the presence of water and with the presence of sunlight. It will evolve what? It will give rise to C2HO plus O2. This was the empirical, and here the CH2O is carbohydrate, which is glucose, a 6 carbon sugar. Okay, Then a milestone contribution to understand the photosynthesis was made by a microbiologist, which is Cornelis von Neil, and who, based on the studies of purple and green bacteria, demonstrate that photosynthesis is essentially a light-dependent reaction in which hydrogen from a suitable oxidized compound reduces to carbon dioxide. He gave this equation. Okay. In green plants, H2O is the hydrogen donor. Okay, and this is something very important for you to note down, that we think that from where the where the O2 has come from, CO2, from CO2 or from H2O. Right? We used to be confused in it. But you need to keep in your mind that we take O2 from the hydrogen only. We don't take O2 from the carbon dioxide, right? Keep it in your mind. And therefore, H2O is the hydrogen donor and it is oxidized to O2. Some organisms do not release O2 during the process of photosynthesis. When H2S instead of it is the hydrogen donor for the purple and the green sulfur bacteria. And this is the correct equation for the process of photosynthesis which was given later, which is 6CO2, like it is written, 6CO2 H12H2O. The presence of light will give glucose, which is C6H12O6. This is our glucose. And do you know the formula of sugar? Sugar is C12H24O11. Okay, and the O2 is released